and hybridization kit compatible with the mainstream sequencing platforms like the MGI, Illumina, Thermofacial, and GeneMind, etc. And besides, we also can provide some reagent for the long-read sequence for the PacBio and the Nanopool. So besides the commercial panel, we can also offer comprehensive customized capture sequencing solutions for the clinical testing, scientific research, and sequencing services. So our headquarters, as well as our R&D and manufacturing center, are located in Nanjing, China. We operate 2 GMP-certified production facilities, including an NGS reagent manufacturing facilities and in-house oligo manufacturing facilities. All the props are synthesized internal instead of being outsourced. This allows us to strictly control product quality and batch consistency, while also ensuring a stable supply and reliable delivery timelines for our customers. You can see here our R&D investment ratio is more than 25%. With a strong R&D compatibility and close interaction with the market, we are able to quickly respond to customer lists and provide cost-effective NGS solutions with consistent performance. Since the company foundation in 2017, the Nanobio has demonstrated strong R&D and innovation ability through numerous inventional patents and software copyrights. Because with the NGS solution, not only the reagent plays an important role in the workflow, but also the informatic analysis pipeline. So we have a lot of the pipeline suitable for our commercial panel. So as I said, Nanobio is focused on the comprehensive series of reagents for targeted enrichment based on the liquid phase hybridization capture technology before sequence, and we also provide the bioinformatic analysis for the sequencing data. And allied with the complete NGS workflow, we offer 4 product lines. The first product line is the NGS reagent, panels, and kits that covers the library preparation and hybrid capture target enrichment processes. The second product line is the prop design and synthesis services, which helps clients to design and order their own prop panels online. And then the third product line is the NGS automation platforms that support fully automated, simple-to-library NGS workflow. And the last product line is the bioinformatics pipelines for sufficient, efficient, and comprehensive analysis of NGS data. So you can see here on the workflow, we focused on the library and the capture and also the analyze. So we see that the library preparation is a very important step in the NGS workflow. So we have developed series of the library preparation kit. So we showed all the modules in this slide. So you can see here for the DNA library preparation, our kit is suitable for both the solid culture fragmentation method and the enzymatic-based fragmentation method. So besides for the regular DNA library preparation, we see that a lot of clients focus on the early cancer screening. They want to detect the DNA methylation level. So you can see here, we also provide the client with the DNA methylation library preparation kit together with the bisulfite conversion module. And then for the RNA, for the transcription detection, we also provide you with the RNA library preparation. And we have several reagents here. And then we have a special kit we call the DNA library RNA co-library preparation kit. This kit is specially designed for the pathogen microbial detection. Because in the clinical samples, maybe in one sample there has the DNA microbial and the RNA microbial. So with this kit, they won't miss some information in the sample. And as I said, our kit are suitable for the mainstream sequencing platforms. You can see here for the MGI platform, we specially developed this kit we call the Universal Circularization Kit version 2. So with this kit, the library can be circular. After that, they can perform the sequencing on the MGI platform. After the library operation, the sample has as a barcode can differ from each other. After that, they can use our panel together with our hybridization system to perform the hybridization. So you can see here we list all the system related to the lab hybridization. So this one and this one, the ES1 and the hybrid capture region, these two are suitable for the traditional 120 NT probes. And these two kinds, they differs only in the buffer, but the panel and the block are the same. And then we give you this one, the Mucala hybrid capture reagent. This is compatible with our patent technology Mucala 1. So the probe with this system is shorter than this two. And I will give you a detailed introduction about this Mucala technology. So we see that on the market, most of the probe have the length 120 NT. And sometimes with this kind of the probe, it's not very suitable for smaller panel because use this one together with the smaller size of the panel, it may be difficult to reach a very high on-target rate. So we developed this Mucala hybrid system. You can see here the probe length range from 20 NT to 100 NT. So the shorter probe will combine with the fragment very fast. This system supports the hybridization only need 1 hour. And how we can make sure about the specificity with the shorter probe. So you can see here we have special design here. After the probe target the fragment, it will also combine with the probe near it. So by this way, we can make sure that the combination will be very fast and fixed. Now, this technology has been used in some, if you are looking for smaller panel, like some used for the MRD detection, early cancer screening, and also some mini panel, and also the pathogen microbiome detection. So so sometimes you can, based on your own requirement, to choose different kind of the probe type, the panel size, and you can make your requirement based on your own need. So besides for the hybridization, we give you a series of the commercial panel we list here. So you can see here, we based on the various hybrid capture system described above, we offer the validated and also the optimized commercial panel for the West series, and we have the series panel type for the newborn screening. Besides for that, we also have can provide some panels for the common genetic disorders like the HLA typing panel for HLA typing. We have the HGB for the thalassemia detection, and besides for that, we have the solid tumor panels for the pan cancer detection, and also for some specialized specific cancer type. So later I will first introduce the panels for the genetic disorder screening first. So on this part, I will first give you the brief introduction about our solution for the genetic disorder screening, and next I will focus on the West panel series and the newborn screening panels. So we say that the next generation sequencing, also called the NGS, offers significant advantages for genetic disorder screening, and because of its high throughput data, NGS can simultaneously analyze hundreds to thousands of genes. It can help us analyze the different kind of variation like the SNVs, indels, CNVs, etc. through the WES or WGS. And now with the technologically advancement, the cost per test has decreased significantly, while the turnaround time has been much shorter compared to the traditional method. These improvements make NGS an increasingly practiced and powerful tool for both clinical genetic disorder testing and large-scale screening applications. Therefore, in response to the market changes and demands, we have developed multi-products for various detection applications. From this table, you can see that we have provide different kind of products for different kind of applications. Like we have the universal DNA library preparation kit for the NIPT solution. And for the universal genetic diagnostics, we give you the 4 kind of the West panel. And each one has the distinguished features. You can select them with your own requirement. And then we have the MGS panel series specially designed for the newborn screening. Besides for the universal genetic disorder diagnostics, we also give you the common genetic problems solution. Like as I said, we have the HLA typing panel version 2. This panel covers 11 HLA genes full length, and it supports very high resolution typing results to the 4th digit. And then we give you the HCP panel. This panel, we are most focused on the hemoglobin-related disease, and we also cover the full length of these genes, so you can use this panel for the carrier screening and also the thalassemia diagnostics. And then we have another one. We have the DMD panel. So beside of the commercial panels, we understand that the customized requirements are diverse, highly diverse. So beside for a commercial panel, we also can provide them with the customization services. They can just provide us with the regions and the genes, the site they are interested in. We can do the customization for them. So okay, let's move on with the detailed introduction with the West panel series. The whole exome sequence refers to the target capture and sequence of all exons of genes. Exon actually makes up 1 to 2% of the genome, but contains 85% of the disease-causing mutations. And compared to the WGS, WES is faster and less cost to perform, while still provide the guidance for identifying genetic disorders. For now, exome sequences is widely used in genetic diseases, typical clinical phenotypes, and rare diseases. Based on the hybridization capture, our company has developed 4 ready-to-use West panel. Each panel has distinguished features and is optimized for different synchro layouts. You can see here from this diagram. The core panel is the basic version, and it's only cover the CDS region. And then we give you another 3 panels. First, the plus panel and the XP panel. It's based on the core panel to do addition. First, the plus panel is the enhanced one based on the basic version, and you can see from here. It adds the whole genome SMP framework can use to detect the chromosome copy number variation, and we also add some the intron regions related to the common gene fusions in solid tumors, and we also have add some MSI site can use for the immune therapy. And then the XP panel is based on the +1 to add more region. First one, you can see here, we add some region related to the IGTR, the immune globulin and the T-cell receptor loci. And then we expand the CLIMA region and also the encode annotation region. So actually, if you want to get more comprehensive and more complete variation information, you can choose this one. it can in the same time get all the information for the germline and also the somatic mutation information. And you can notice that we have a mini one, which have a more less covered region. It only cover like the 16 megabase of the genome region. But in this panel, we have special consideration because we understand that on the market, most of some of the clients focused only in the inherited disease detection. But if we choose the core panel or the other two, it will have a very high data volume and it will be very high cost and it's very comprehensive. It's very complex about the bioinformatics analysis. So we give you this one, mini one. It's most we involved the genes related to a clinical inherited disease, and all the exome panel we list here can be used for the newborn screening, carrier screening, genetic diagnostics, and also the research user needs. So here I would like to emphasize the highlights for our exome panel, the West panel. So first you can see here the exome panel is designed based on the Human References Genome Version HG38, and it also provides bat file based on the HG90 to assist users in coordinate conversion. The HG19 intervals converted from the HG38 have undergone manual correction to prevent the accuracy of the abnormal regions. You can see from this figure here, the red region from the vector A and vector I it's not the true target coverage. Only our core panel give the accurate annotation with this region. Besides, you can see, besides an important highlight is that we use a smaller panel size, but we get a similar coverage. How do we achieve that? You can see here, we just get rid of the excessive intron region from the West panel. So by this way, we can just make sure that we have the very comprehensive coverage, but we do not have the get more covered get a higher panel size to increase the sequencing data. And then here we also make the combination to show that we have the enhanced coverage of the important variation site. We make the combination with our core panel together with the vector A because these two products has the similar coverage target region. And you can see from the figure on the left. Every time we will find some low coverage losses from the West result. And then you can see here for the important losses, important region, we have less region with the low coverage depths compared to the vector A, so we can get more information. And then we also checked with our coverage uniformity compared to other vectors. Use the special region RB1 exon 1 with the high GC content and the BRCA1 exon 16 with the shorter length. You can see here, compared to the vector, our core panel can have a very uniform coverage in this other region, the high GC content and the short length region. Besides, you can see here that the vector B and the vector D have some poor performance in this low C. So except for the highlight we marked here, we would like to talk more about the biggest one, the XP panel, and the smallest one, the mini WES. So here is some information about the XP panel. So the four table is a presentation of the XP panel's expanding coverage of the long coding CLIVA variations compared to our core panel and the Vidar A. You can see that for several clinical significant region, there are less low coverage loss in the XP panel. And then we introduced that with the XP panel has enhanced coverage of the encoding along coding regions. And from this table, you can see here, our XP panel compared to these three, we have a more covered average level with the encoding region. And also we make the comparison here compared to the XP panel with our core panel. The blue one, the blue represents the XP panel and the green represents the core panel. We can clearly see that the blue area is larger. That is that the XP panel has a higher coverage of the encoding. In addition to the XP panel, I would like to specially introduce the mini-wise panel to you. As I introduced the previous, the mini-wise panel focused on the exon associated with the clinical inherited diseases with PrEP coverage spanning 60 megabase genome regions and involving more than 5,600 clinical significant genes and encompassing over 4,000 genetic disorders. And compared to the whole exome panel, the mini panel can rapidly detect pathogenic genetic variations with the target genes, thereby reducing the data analyze time and minimize the turnaround time. Besides, it aims to only include the clinical significant regions specifically related to the inherited disease, and it can effectively reduce the sequencing cost. So how the mini panel achieve high efficiency and low cost? First of all, from the bar chart on the left, when it comes to the sequencing depth to the 100 times or 200 times, the amount of the data required by the mini panel is less than half that of the core panel. By saving the amount of the raw data, we achieve the cost saving. And also you can see from the figure on the right, the mini panel covers a smaller region, only account for the 38% of the core panel. The smaller the panel, the less cost is required. Here we also, as I said, we have the pipeline for all commercial panels. We have developed the analyze pipeline, and the software can help us generate the analyze report. So here we list the demo report of the mini-wise panel. Typically, the report includes the data quality control parameter, and we also will give a summary of the analyze result, and then it will be the detailed variation information. The variations are classified according to the ACMG. Then we give you this table. This table summarizes the parameters comparison among our 4 exome panel. So you can, so from this table you can know more information, and it will help you to choose which products is most suitable for your requirement. So first you can see here the PrEP covered area of the first 3 panels is relatively larger and reach more than 40 megabase, which is called the whole exome panel. And the coverage region of the mini one is the smallest one. And then you can see here all the on-target rate and all the uniformity parameter looks very good, but because that the mini-wise is the smallest one, so with the 10 gigabit the sequence zero data, it can achieve the more than 330 times of the average sequence depth. And then although that this panel has different coverage coverage region, but with 10 gigabits of raw data, all these three panels can achieve a very similar average depth. So sometimes you maybe these three panels won't have a big difference with the sequencing cost. And then you can see here for the XP panel and the plus panel, we have add more region, like the SMP framework can use for the copy number variation, and also some the intron region related to the fusion detection, and also MSI site. So if you are more focused on the inherited disease, you can choose this one. And if you want to get more information, you can choose the XP panel. And next, we give you the workflow and the reagent, detailed solution about the reagent for the West panel detection. So you can use a gDNA or FIP sample to perform the extraction. After that, you can together with our library preparation kit to perform the library preparation. You can choose the different adapters suitable for different sequencing platforms. After that, you can choose the panel suitable for your own requirement to perform the hybridization. After the sequencing, we can help you to analyze the result and generate the analyzed report. And you can see here, we also list some of the requirement data volume here. For the mini one, we recommend 3 gigabits of the raw data. And for the other three, we recommend the 10 gigabit of the raw data to achieve like the every sequence depth 150 times. And here, the recommended data volume we list here is just for the germline mutation. If you want to use the West panel to perform the to detect the somatic mutation, we recommend a higher sequencing depth, like the 500 times. So you also need to increase the sequencing sequencing data volume. So this part will be some information about West panel, and next part will be the introduction of the newborn screening panels. Newborn screening is part of the public health systems, aiming to save lives and improve the life quality of the newborns with treatable disorders. Newborn screening helps to identify genetic conditions that may not be apparent at birth, but would lead to some health problems later in life. So this page is the overview of the evolution of the newborn genetic screening. In the first time, the hair-pink dried blood spot is used for the screening of the PKU, and then the MS and the integrated molecular diagnostics can use for some such kind of the requirements. Now, in the 2020s, NGS has shifted screening from the phenotype drive to the genotype drives module and covers over 100 treatable genetic disorders. Each iteration has aimed to protect newborns' health earlier and more precisely. This slide covers the key global progress in NGS-based newborn screening, genomic screening. Chinese newborn screening with a targeted sequencing study validated target gene screening and shaped 2022 local expert guidance, while the US and the Australian studies showed GMBS as the first titer tool can boost the detection rate and has reliable timelines and strong parent acceptance, backing its clinical healthy use. So we see that newborn screening is one of the most successful programs in the global public health system. It identifies intervalable diseases before the some symptoms appear and significantly improving the outcomes. However, the traditional screening technologies such as immunoassays, many rely on the biochemical indicators. They struggle to cover diseases with strong genetic heterogeneity or not reliable biochemistry markers and cannot explain results like the false positive and false negative. Those false positive results will cause the significant pressure on the families and the individuals. So as the numbers of the screenable diseases continues to expand, the traditional method facing the trouble, including the insufficient sensitivities, limited throughput, and the low positive value. So now the newborn genetic screening use technology to detect the disease-associated pathogenic genes, and the positive variations identified can be verified by single sequencers. MGS not only eliminate the TMBS positive caused by the high-risk factors, but also effectively compensate for the TMBS missed detection. Currently, we offer we offer multiple West products that can be applied for the newborn screening. However, in such kind of a situation, the West will requires a relatively high sequencing cost, and the bioinformatic analysis also more complex. So therefore, we have developed the NBGS panels, which is specially designed for the newborn genetic screening applications. Here you can see here the NBGS Premium, the panel variant 1, the largest one, targets more than 300 clinical significant and actionable hereditary conditions, and it covers more than 280 genes. The prop design target multiple variations, including the SMP, indel, and the CNV, etc. And the panel size is 1.3 megabase regions of the human genome. It's not such large, and it's a very high sequence data volume. So for your reference, this is the disorders and the related gene list of our panels. We selected these disorders and the genes according to not only just our Chinese specialized consensus, we also take the WHO, the Wilson-Janger criteria, and we also refers to the largest rare diseases database, the alpha 100. This is the list. We list the disorders, and we also list the genes related to the disorders. Maybe you can see that there is some overlap with the genes list, because sometimes maybe for different disorders there are also they own the similar drive genes. Here is a highlight of our products during the design processes to balance the detection result with the cost considerations. We did not include the full-length sequences of the disease-associated genes, but instead we focused on the pathogenic related variations. Thus, we can cut down the size of the panels, not allow us to do the deeper sequence to get more information like the CNV, that kind of variation. So because also we know that the CMV detection is very important in the NGS, but it has some sometimes if we cannot get a very high sequence depth, we only get very precious information. So for the CMV detection, we designed the SMP backbone, perhaps cover the intergenic and the intronic regions to improve the resolution of the CMV interpretation. And we also optimized the depth coverage of the K genes by supplementing the critical intron region and the UTR regions outside the CDS to enhance the variation detection sensitivity. And then beside of the premium one, we also give you the three smaller one, because when we develop this panel, we take the 90% of the inherited disease around the world, but in different country, in different area, the client may be focused on the different kind of the disease. So we also, besides for the largest one, we also give you the smaller one, and you can check with the gene list and also the disorder list to see if the which one is better for you to choose. Also that we understand that different clients have different requirements. So once you check with the list and you see some important regions that is not involved in this panel, we can do the customization for you. You can give us the region you are interested in, and also we will check with the database to see check with the pathogenic site, and we will customize the prep based on the present strategy to help you to customize the panels and spec in with the commercial one. And also for the MBGS panels, we have already developed the analyze pipeline, so we can help you to generate the demo report, analyze report. Sorry. So besides for the panels can use for the early cancer, for the newborn screening, we see that we have do some market research, and we found that some of the carrier screening genes are also included in our newborn screening panels. Therefore, we have specially provided the list of genes covered by the core panel, the middle one, for your reference. You can check with the genes, see if the genes are those you are cared in the carrier screening. You can check with the gene and also the responding coverage site, and then if the panel can, you see that the panel can cover all the information you want. It can not only used for the newborn screening, but it also can use for the carrier screening. Besides for the basic information, we also show you some performance with our panels. We use some standard to do the validation with because that in this panel we also covers the HTTP and DMD related the thalassemia and DMD related genes, just not the full length. But if you have requirement for the full length of these genes, we can provide you a commercial panel. You can replace them with a separate prep with for the thalassemia and the DMD, and you can get the full information about the full length of the genes. Here we marked we give you the capture performance with the DMD, gDNA, and you can see here with we show you the parameter like the on-target rate and the uniformity on both the Illumina and MGI platform. You can see here all the parameter looks very excellent here. Besides for the capture performance, we also give you the validation result. This one is the result for the references, and this one is the observed information. You can see here, beside not only for the heterozygous deletion, and also some like the zygote deletion result, we can detect that by analyze the sequence depths. And also for this is the deletion result, and this is the amplification result. We can just analyze such kind of information, and then we will put them in the demo report. So here is the example for the NBGS analyze report. We also will list the QC information, the summary of the analyzed result, and we will give the detailed information for the variation interpretation. So because we have our software, we have the pipeline we develop by ourselves. So normally you can it will be very flexible for you to generate the report. You can put the information you are more focused in the report and to show you to the client. And then we give you the workflow. So maybe for the newborn screening, you mostly use the blood sample to the GDA sample to do the test. And we also provide you with the reagent here and also the panel and the recombinant data volume for the newborn screening, because as I said, some variation like the CMV is very important, and it may need deeper sequence depths. So for the newborn screening, we recommend the average sequence depths around 200 times to 400 times. And here we give you the recombinant data volume based on the 400 times sequence depths. You can adjust that by your own requirement. And these two parts will be some solution, the West panel and the newborn screening panel for the inherited disease detection. Besides for that, we also have the series of the panels developed for the oncology detection and also for the early cancer screening. So from this slide, you can see here for the oncology, there are a lot of the biomarkers like the CMV, indel, fusion, SMV, etc. These are mostly used in the cancer panel, and also the TMB, MSI can use for the therapy, and the HRD can use for the pyrrole inhibitor, and the methylation can use for the early cancer screening. Based on the biomarkers we list here, we developed a series of the commercial panel. So this part will be some the panels for the oncology detection. So you can see here they can be divided into two parts. One part is focused on the solid tumor solution, and another part is focused on the blood cancer. For the solid tumor, we give you the 4 types of the panels can use for the pan-cancer detection. The plus panel, OncoPlus Fusion 3, is the biggest one. Can use for detect a lot of information like the SMV, indel, CMV, fusion, and also MSI and TMB. And then this OncoForEdit panel is a special one, because all the other panels are based on the DNA template to design the prop. But the OncoForEdit, we specially design this based on the RNA sample, because sometimes for the RNA sample, if you use because sometimes for the fusion detection, at most suitable if you use the RNA sample to get the to check with if the fusion happened and if it has been transcribed to the mRNA and have the function with the protein. So sometimes together with these two plus Fusion 3 and OncoForEdit, you can get a very comprehensive information of the different kind of variation for the pan-cancer detection. And then we give you the 2 smaller one, also designed for the pan-cancer detection. The long CT panel is specially designed for the liquid biopsies. Can use a cell sample to detect the different kind of variation. And also in this panel is small one, but we also involved some MSI site can use for the immunotherapy, the chemistry therapy. And then the Mucala hotspot panel is a very smaller one. It's very small, the smallest one. This panel is designed based on the Mucala technology I introduced in the beginning, and it can also used for the liquid biopsies and also sometimes like the MRD detection because the panel size is very small one. Besides for the panels designed for the pan-cancer detection, we also have 3 specially panels. One is the lung cancer panel. This panel we involved the genes and the variations related to the different kind of the therapy like the immunotherapy. And then we give you the HRR panel and together with the high sleep ultra panel. The HRR panel used for the HRR pathway gene mutation detection and the high sleep ultra panel used for the HRD detection. So together with these 2 panels, you can just use for some application for the HRD detection and can use for the PARP inhibitor therapy information. And besides for the solid tumor solution, we also give you 2 panel designed for the blood cancer. The NanoHema panel 2, version 2 is a special one because this panel we have a separate tubes. One tube is based on the DNA sample and one tube is based on the RNA sample. The DNA tube can use for the traditional the normal variation, and the RNA tube can use for the RNA expression level and also the fusion detection. So we recommend you together with this panel, you can just use for the different kind of the blood cancer detection. And then we give you the Mucala AML panel. This panel also based on the Mucala technology and special used for the AML MRD detection. Then we give you the workflow. So for the panels designed based on the RNA, you maybe have to use to reverse the RNA to DNA first and conduct the library later. But now we already have launched recently we launched the RNA easy library preparation. You don't need to 2 kit, only one. It's okay with the RNA sample. And if the sample type is a DNA, you can just go to these following steps for the library preparation and hybridization. Also, for all the commercial panel we list here, we already have the analyzed pipeline. And if you have some requirement for the customization services, you can let us know. Besides for the panel, we can also help you to customize the pipeline, analyze pipeline, and help you to generate the report. So this will be some information, some products for the oncology detection, and also we have some solution for the early cancer screening based on the DNA methylation detection. We have 2 kits, and these 2 kits based on different kind of method. The Mucala EMS panel version 1 is based on the conversion method. We can use we have to use the enzyme or the bisulfite to convert the unmethylated cytosine into urals. So by this way, you can differ the unmethylated cytosine to the methylated one. So based on that, we give you the Mucala EMS panel version 1. And then we understand that sometimes for the clinical sample, maybe you won't be able to get a very high initial input. And also sometimes for the early cancer screening, we use the cfDNA to do that. So maybe sometimes the bisulfite will cause some DNA damage, and also sometimes for the early cancer screening, the client want to combine the methylation with the mutation together. So we give you this one, Mucala EMS. Mucala FS-EMS Plus panel. This panel is not based on the conversion method, but we use a special enzyme. We call the methylation sensitivity restriction enzyme. This enzyme we call MSRE. This enzyme works like this. So first, it will recognize the special site, the enzyme site first, and after that, it will check about the CpG state here, If the CPG is unmasked one, it will clip here. And then if the CPG here is the masked one, it will just remain to the initial state and it won't be clipped here. So we have used this technology. We have used this enzyme together with our Mucala technology to develop this Mucala FSEMS plus panel. So this panel can just use for some early cancer screening, and it can combine with the other Mucala mutation panel together to use for to simultaneously detect the methylation and mutation. And it can also use the long for the DNA methylation detection. And then we give you this workflow. So if you choose the EMS one, you have to perform the conversion step, and then you perform the hybridization. If you choose the MSRE, this technology, you can just go to perform lab preparation, and during the lab preparation, we will provide the enzyme to you use that together with our pen together. And after the sequence, we have the pipeline. And especially for the FSEMS plus panel, this is a very special one. So we can provide you the Docker to help you analyze. And we will output the DNA methylation level of every CPG site. So this part will be some solution and commercial panel for the oncology detection and early cancer screening. Next part will be some information about the deep customization services. As I said, that different client focus on different region. So actually, we have this website, the online design platform. So it's open source. You can use. We can provide you the guidance. You can use a website to design product by yourself, or you can just provide the information or the region or the specialist site to us, the batch file to us. We can help you to design the product. And after the design, after you make sure that you want to order the product from us, we will synthesize that for you. So you can choose that you can choose different product, like the traditional. If the panel size is very large, you can choose the traditional one. If the panel size is more than 100 kilobase, we will recommend you to choose the traditional 120 NT. And if the panel size is very small, like it only like the 12 kilobase, we will recommend you to use the Mucala technology because it's more more suitable to use this technology to achieve like some on-target rate more than 70%. And if you want to do some high sequence depths, you can use this one with a high on-target rate. You can just save the sequence data volume. So we have our own synthesized for manufacturing facility for the product, and our synthesized strategy is that each product synthesized step is independently synthesized, and it will inspected for the quantity. So during the QC part, after we synthesize probe, we will do some QC. In the QC part, we will check with every probe with the concentration, and make sure that when we pulling the probe together, they have the consistent concentration to ensure about the temperature uniformity. And then we will use the MS to check with the molecular quantity to make sure that there won't be something wrong with the product. And then if the client have some requirement, we can also perform the NGS QC. We use the hybridization experiment to check with the capture parameter like the on-target rate and the uniformity, GC values, etc. And then we will deliver the panel to you. So this was some the customization services for prep design, and I would like to show some performance with the personalized prep customization services. So you can see here we check with the concentration from the different with the different quantities. You can see here there is involved in the different panels. There are different numbers of prep, and you can see here the all the concentration is with a very similar region, and the CV is less than 10%. Well, maybe some vendor also can provide you with the synthesized services, but on the market, many of the vendor can just get the CV less than 50%. So in this part, we do better than them. And then we also check with we also show you some about the number about the prep present. So the prep present means that refers to the proportion of the prep regions with the average sequence depth greater than 5 times when the overall average depth is 200 times. And we from the different from the selected customer preps from the different batches, you can see here the prep present rate is 99.9% or higher. So also that this is some performance with the customized panels, and also we check with some information, check with some performance with our commercial panels. Because sometimes when we choose products, we not check with the performance, maybe the combination to you, but we also have to know that consistent and the stable performance will be stable or not, because in the clinical applications, the stable of the reagent is also very important. It will help you to reduce some the cost and to get a very stable result. So here we use our OncoPlus Vision 3, the pan-cancer panel, to do the validation, and you can see that from different batch, the target coverage is very consistent with each other. Some performance with our prep synthesize. So this part will be the customization services, and last part will be the end-to-end fully automated NGS solution. So for the wet lab, for the hybridization and maybe the lab preparation, for some application with very high, with a lot of sample size, only by manual will involve a lot of the fault and will just not be easy to perform all the experiments. So we developed a lot of the fully automated NGS workstations. We list here there are 3 kinds, and each kind have a different feature and support for different kind of scenarios. So first we give you the Nanodigmbio 16. This is the smallest one, and you can see here about the channels. It has a fixed 8 channels, and for the lab preparation, it can perform the 16 samples one time, and it can go through all the steps within 3 hours. And then if you want to use this machine to perform the hybridization for one time, you can perform the 8 reaction. So for our hybridization, if you choose the traditional one, the ES, the traditional one, the traditional prep panels, we can provide you with a ES panel, ES hybridization system reagent. It can support the hybridization in one run. It can support 1 to 12 samples in one pooling. So if you perform the 12 samples in one pooling to perform the hybridization in one time, you can just perform the hybridization for 96 samples in 6 hours. So this workstation is more suitable for some small scales of the sample size. And then we give you another tool, the D4. The D4 is very special one because it has the 4 independent channels. And you can see here it can support the library one run in one run it can support 1 to 16 samples to perform the library operation. And for the hybridization, it can support 1 to 8 reaction for hybridization. And then this D4 is most suitable for if the sample amount is not so stable. So you can choose this one. And then we'll give you a largest one, the 48. The 48 has a high throughput. It has the fixed 24 channels. In one run, you can perform the 48 library preparation. And it can perform and for the hybridization in one run, it can perform 24 reaction. So if the sample size is very large, you can choose this one. So besides for the workstation, we also have developed the plate kit suitable for the manual experiment and also the workstation platform. In the traditional tube, before we just divide the reagent into the tube. So the tube, if you use the tube to perform the experiment, it may have some possible that you have misuse of the reagent. And if you want to use the workstation to perform the experiment, if you use the tube reagent, you have to prepare the reagent and it will cost a lot of time. And because we see that the library preparation and hybridization will take a long time, and if you use the tube reagent, you will just lose some reagent during the experiment. So we developed this plate kit. So in this kit, we divide the reagent into every column. Every column contains the reagent support for one reaction. So by this way, you don't need to waste so long time to prepare the reagent, and it won't lose some reagent. This is the example for the plate kit for the Nano 16. This is a library preparation kit, and this one is a hybridization kit. And also for the 48, because it has a fixed 24 channels, we also have a specially plate kit suitable for the different machines. Besides for the design, we give you some performance to get use our workstation. This is we use the Nano 48 to perform the library preparation. You can check with the library yard and from different batches, and also the GC and also the fragment size. This is for the library preparation, and for the hybridization, we use for the hybridization, we use a small panel, a lung cancer panel, to perform the experiment, and you can see that on target rate, and the mapping rate looks very good. Also the uniformity parameter, and from different batches, the performance is very stable. So it's just an example for some performance with our automation system. So you can see from the beginning until here, we provide the we can provide the end-to-end fully automated NGS solution. We can provide you with the library preparation kit. We can provide you with a panel suitable for different kind of applications, and also the capture reagent. We have the automation system, and we also have the analyze software. So we can just provide you the fully automated NGS solution to enhance the efficiency in precision medicine, that kind of the situation. So this will be the total solution, all the information I want to introduce to you today. So if you have any question, you can let me know. We can discuss about that.
Hi, Judy. We have one question in the chat box. Can you please share the diagnostic sensitivity and specificity of your newborn screening panel?
Newborn screening?
Yeah.
About the workflow?
No, sensitivity and specificity.
You mean the result here?
The diagnosis sensitivity. Yeah.
Panel sensitivity?
Yeah.
Okay, which we don't need some the parameter about the sensitivity and the positivity, because for if we want to check some information like that, we have to collect a lot of the clinical samples to check with about the parameter. But because we are completing the biotechnology company, we have a limitation. So we cannot collect so many kind of the clinical samples and to provide some information to you. We are sorry about that.
Dạ, thì ở đây em thấy có một câu hỏi từ Itachi, nickname là Itachi. Thì hiện tại thì bạn Judy bạn chưa có cái thông tin về lâm sàng do bị giới hạn bởi cái việc mà thu mẫu á. Nên là nếu mà ví dụ như là mọi người có cái lượng mẫu và muốn kiểm tra xem về cái độ lâm sàng thì bên em vẫn có thể tiến hành hỗ trợ cái chương trình demo cho khách hàng ạ. Thì không biết các anh chị còn có câu hỏi nào thêm không ạ? Dạ, mời bạn Nguyễn Phương Mai.
Chị đi ra ngoài mà nói.
Hello, so I have two questions. The first question is about the Nexome XP panel. So I want to ask about the CNV detecting ability of that panel, because we have ran that panel in our department, and some CNV appear to be not correct, because like several patients carry the same CNV that is pathogenic. So I'm afraid that the ability of CNV detection of that panel is not very good. And second, I want to ask if you have some panel that can detect the gene nucleotide repeat extension, like in FMR1 gene or AR gene or other genes relating to neural disorder. So I have two questions.
Okay.
Thank you.
So, yes, thank you. So for your first question, you checked with, so you have performed the experiment with our X panel. How about the on-target rate and some uniformity parameter? Does that looks good? And also sometimes you have checked with the something wrong with the CMV did not be detected. Have you checked the sequence depth in this region?
Yeah, I have checked the IGV image, and the coverage of the prop in those CNV region might not be reliable enough. But is there any kind of like quality control, or like I don't know, like like some way to call the CNV correctly, because it's still quite unreliable, the CNV result of that test. But we also detect some CNV, especially the exome deletion or duplication, that we also confirmed by Sanger sequencing that is right, that is correct. But there are a lot of CNVs that is really unreliable. So I think the CNV calling of that test is kind of not really good enough for that, not meet our demand,
Okay, so so the all the CNV you have bad performance are in the CDAS region or some other region?
No, actually there are some CNV that is pretty large, like more than 10 megabase. Yeah, and it expand the many regions. Yeah.
Okay, so we can help you to check with something similar with this CMV with this region, and you want to optimize for the performance with this region, we can help you to add some implement some prop in this region to help you get better performance with the CMV detection result. And the second question is about that if you want to check with if we have special panels for some gene.
For the G-nucleotide repeat extension, like in the FMR1 gene, or in the AR gene, or in other genes relating to neural disorder.
SMA?
No, not SMA. I see that you have a panel for SMA and DMD, but other genes
Okay.
like DMPK, the G-nucleotide repeat expansion.
Okay, we can collect your requirement, and we will check with the list to see if there is a commercial panel can meet your requirement. If not, we can also help you to customize the panel for you. Okay.
Okay, thank you.
Dạ không biết mình còn ai có câu hỏi gì không ạ?
Can I ask a question?
Yeah, okay.
Sure.
For early cancer screening, so what kind of cancer can be used this group with a kit? And for minimal residual disease assessment, do we need to do 2 times for comparison using the methylation profile?
Okay, so first for the early cancer screening, we have the as I said, we have 2 commercial panel, and one is the EMS one. The EMS one we have covers the 9 cancer type.
Yeah.
Like this, you can check with the list we marked here. We can use the Mucala EMS panel to detect the lung cancer, the liver cancer, such kind of the cancer type, and we also list the biomarkers related to the lung cancer, different kind of the cancer type. So this is the cancer related to the covered by the Mucala EMS panel, and then for another one based on the MSRE. This method, we give you the FS-EMS plus panel. In this panel, we cover 10 cancer type, and we also list the biomarkers related to different kind of the cancer. You can check here, and we can also provide the brochure, and we can provide the gene list to you.
So how long can detect for early detection before we can detect in clinic?
Yeah, I understand your concern. So before you use the panel in the clinical situation, we have to build a module first. So because in the panel and in the panel list here, we involved a lot of CPG sites and we involved a lot of the cancer type. But we based on some the CPG site select some CPG site from the like the FDA approved and some literature report. So you have to collect some clinical sample like the hundreds of the clinical sample to use our panel to build a module first. We can help you to do the test, to do the experiment, and we build the module first, and you can check with the build the modules to select the CPG size, especially for different kind of the cancer type, about the level, about different kind of the parameter first, and then you can use that to some clinical situation. So the turnaround time will based on the first you have to collect enough sample and you have to perform the experiment. Also, we can provide your doctor with the analyze. And after you have conducted the research and you have built a module, you also have to collect some sample to check, to evaluate the module you built now is correct or not. So it may take some time, but it also depends on the how long can you complete the total experiment and analyze and the validation step.
And the last question for minimal residual disease assessment. So do we need to do 2 times, for example, before treatment and after treatment after some time? So for the comparison of the methylation profile.
You mean that if for the MR detection, you want to combine the methylation and detection together?
Yes. Do we need do we need only 1 time for assessment, or 2 times comparison before and after treatment?
So if you choose our Mucala system based on the MSRE technology, you can just perform 1 time of the experiment. But also, if you want to use the methylation, in the MRD detection, you also have to build the module. And but if you choose the full screen system, you can just perform one experiment. This is the traditional method. You have to perform two round of the experiment. But with this one, you only need one time of the total experiment.
Yes, thank you very much.
Dạ không biết là bên mình còn ai đã hỏi không ạ?
Trong chat box có câu hỏi kìa chị Vân ơi.
Ok. Hi Judy, you have one question that can NPS using the blood from the dry spots, DNA extraction from the dry spot, and the
You mean
the
that
quality,
for
the quality of the DNA input?
You mean that for the early cancer screening, if you use the blood spot, you want to
newborn screening, we will use the dry spot, dry blood spot. We can extraction the DNA from that, right?
Yes, you can use that. You can use that, and after you do the extraction, you have to check with the concentration and about the nucleic acid quantity. And then
the question about the input requirement for the
so
for the sample.
For normally for our kit, our 2 kit can support the input rate from the 5 nanogram to 500 nanogram. And for such kind of situation, for the early cancer screening, we recommend the input like the 5 nanogram or the 100 nanogram about the input. It can get output the more accurate detection result. But if something if you something wrong with the extraction or the about the sample and the collection, you won't be able to get very high input to the 15 nanogram. So less than that is okay. Just not less than 5 nanogram.
Dạ không biết thì câu trả lời có làm hài lòng bạn Phan Điệp chưa ạ? Dạ không biết là bên mình còn anh chị nào có câu hỏi gì không ạ? Dạ, nếu không thì em cũng xin rất là cảm ơn các anh chị đã dành thời gian để mà tham gia cái buổi hội thảo ngày hôm nay. Thì cũng kính chúc anh chị sức khỏe. We have one more question.
We have one more question.
Yeah. In the one run, do we have the requirement for the at least how many sample and the maximum how many sample for one run? Is it in the newborn screening panel? Dạ chị Mùi ơi, có phải là ý chị đang hỏi về newborn screening panel không ạ? Hay là cho panel nào ạ?
You mean that for one run, how the minimum of the sample library?
The question of the customer is not clear because she asked us about how many sample we can run maximum and minimum. But I not yet know that her question is about the newborn screening or the exam panel or any panel else.
So actually for the requirement for it will depend on the sequencing platform, the chip, the volume. So we have listed all the requirement data volume here. You can calculate them first to see which kind of the platform, which kind of chip can support for how many about the sample size. Because if for the west panel, you can see that for the west panel, for the core panel, KSP panel, we recommend 10 gigabits of the data volume. If the chip is not big enough for such kind of the very high sample input, so it won't be support such kind of situation. We don't have the limitation. We don't have the limitation about the maximum of the. It will depend on the chip, the maximum.
Dạ chị Mùi ơi, thì cái mục mẻ chạy ở đây á, cái dung lượng yêu cầu nó sẽ phụ thuộc vào cái từng loại panel, tại vì mỗi panel nó sẽ có panel size riêng và từ đó thì mình có thể chọn cái sequencing disk nó khác nhau, coverage disk khác nhau, thì bên em cũng sẽ hỗ trợ tính cho mình cái số lượng, cái dung lượng mà tối đa là có thể chạy bao nhiêu mẫu. Có một số ứng dụng nó có thể chạy chung với nhau, nếu mà chạy trên cùng một flow cell như là NIPT thì vẫn có thể chạy chung với BCTA, hay là cái ung thư có thể chạy chung với web khi mà họ chạy trùng, các ứng dụng đó chạy trùng trên một cái loại flow cell ấy ạ. We also have one question from Miss Nga. The newborn screening test have the IVD?
We don't have the IVD now, but we are working on that kind of work to say maybe in the future we will get some the IVD certification.
Dạ, chị Nga, hiện tại thì cái newborn screening của bên Nanodim thì chưa có cái IVD test, nhưng mà bên em cũng có một cái hãng khác cũng newborn screening và có IVD ạ. Nếu mà chị cần thêm thông tin thì bên em sẽ liên hệ. Hi, Mr. Lin, I see you raise your hand. You can open your mic and write a question.
Hi Judy. I came a bit late, so I just want to check about the Naxon Mini. Will this cover like 25 about the Naxon XP? Can it be used for both germline and pan cancer screening? And how about the sequencing depth? Do you recommend?
For the XP panel, the sorry.
The mini one. The mini one.
The mini one. So for the mini one, if we use the mini one for the germline mutation because the mini panel, the N-exome mini panel is special designed for the inherited disease. If you want to use that panel to check with some the germline mutation, we recommend like the 3 gigabits of the data volume to achieve like the average sequence depth around from the 100 times to 200 times. And if you want to use the XP panel, the N-exome XP panel to detect the somatic mutation, we will recommend to increase the data, the sequencing average depth to like the 500 times. So in this way, you have to like get the raw data around the 50 gigabits to get the germline mutation information. But if you are only focused on the germline mutation information, you can just perform the 10 gigabit of the sequencing data volume to achieve like the sequencing depth 150 times. We have the demo result for that.
Oh yeah, thank you.
Hi, Judy. We also have a question from Mr. Khoa. The early cancer detection can detect the cancer the early how many years?
How many years?
Yeah.
The early cancer screening, because the DNA methylation is a very important biomarker. It often happens like before the cancer the risk, like I think maybe 3 to 5 years. I'm not so sure about this information. I think it will related to different kind of the cancer type. We have to check this information later, and I will feedback this information to you. I'm not so sure. Yeah. I think it will related to different kind of the cancer type, and also related different of the patient.
Dạ thầy Khoa ơi, thì ở đây thì bạn Judy trả lời là bạn chưa có chắc, nhưng mà theo cái kinh nghiệm và trong cái giới hạn hiểu biết của em á, thì thường cái early detection này nè, trước khi chúng ta làm á, thì chúng ta sẽ làm cái profiling oncology cho từng bệnh nhân trước, và sau đó chúng ta sẽ chọn cái marker, và sau đó chúng ta sẽ theo dõi MRD. Thì thường cái quá trình theo dõi MRD này nó sẽ phát hiện tỷ lệ ung thư mà nó sẽ trước là 6 tháng cho đến 1 năm. Thì có một số hãng và một số panel thì nó cũng đưa ra cái nghiên cứu về trên mặt lâm sàng với những cái paper, thì em thấy đa phần là sẽ là rơi vào khoảng 6 tháng nha. Còn về thực tế thì bên mình cũng chưa có tiến hành nhiều lắm.
Rồi, cảm ơn em.
Dạ, đó là những cái paper em đọc được thôi ạ.
Bởi vì anh cũng có đọc được một cái tin, nó không phải là bài báo khoa học công bố, mà là chỉ là cái tin trên paper thôi, thì là có thể phát hiện sớm tới hàng chục năm, nhưng mà có lẽ là cái thông tin này cũng cần phải kiểm chứng lại.
Dạ, thì cái thông tin mà em được chia sẻ từ hãng á, thì họ nói là khoảng 6 tháng đến 1 năm thì tốt hơn là sẽ làm lại, do cái việc methylation hóa nó cũng sẽ thay đổi. Dạ.
Cảm ơn em.
Dạ, không biết các anh chị còn ai có câu hỏi không ạ? Dạ, nếu chúng ta không còn câu hỏi nữa thì chúng ta sẽ kết thúc cái buổi seminar online tại đây. Một lần nữa RIVN cùng với hãng Nanodim cũng trân trọng cảm ơn các vị khách mời đã dành thời gian quý báu của mình để mà tham gia. Hẹn các anh chị vào những cái seminar online tiếp theo ạ. Em cảm ơn ạ. Hi Judy, we end the online seminar now.
Okay. So thank you for your time, and thank you for your invite for me to do the presentation. So maybe later Miss Tuan will keep in touch with all you and to collect your requirement. And if you have any question you want to discuss, and you can tell her, and we can collect the information and feedback to you.